Hi there, it's Orna, and I actually wanted to show you how I modeled the Sirtuin pathway and how it essentially affects Alzheimer's pathology. So I go over Alzheimer's pathology in my article, which I'll link in the description, but we're going to look at how Sirtuin 1, which is essentially a nuclear sirtuin, and sirtuins basically regulate cellular homeostasis, and they also are a histone deacetylase. And I explained that more in depth in my article, but TLDR, it makes sure which genes will actually become proteins via the central dogma and which genes will not. So it is an epigenetic modifier, and this controls various aspects of Alzheimer's pathology. We first looked at retinoic acid receptor, and this is specifically the receptor beta. It is a transcription factor, so it basically controls the rate of change of information throughout the transcription process. And transcription is whenever you translate gene, so DNA, to mRNA, and mRNA goes to becomes tRNA, which is makes up the protein. So it's just the precursor. And um, this this also controls and activates the specific enzyme called ADAM10. And the increase of ADAM10 actually leads to neurogenesis because um, secretases, which are alpha secretase, gamma secretase, and beta secretase, they basically cut up the amyloid beta precursor protein into the small fragments which make up the plaques. However, alpha secretase doesn't actually make the plaques because whenever it cuts it, it's not as sticky as it is with gamma secretase and beta secretase. So whenever we cut up these plaques, they don't actually occur. And then if we look at so that means that this doesn't actually prevent the synaptic potentials from occurring, and neurogenesis does occur. Now we go on to peroxisome proliferator activated receptor, gamma coactivator 1 alpha, which is another transcription factor, and this is activated by the sirtuin. And this basically allows for the mitigation of reactive oxygen species, which I also talked about in another article. And this is basically a promoter for the cellular senescence phenotype. So this contributes significantly to autophagy in the brain, and it leads to a decrease in neurodegeneration. Essentially, it cleans up your brain. And it also inhibits nuclear factor kappa B, which are present in microglia, and essentially this promotes these immune cells from in functioning. However, with more nuclear factor kappa B, the microglia become the sort of wild type, and they basically basically become senescent cells or zombie cells that start killing off and excreting enzymes that kind of just muck up the whole system of the brain. So we inhibit this so that it's not as mucked up. Then we also activate fork head box O3, and this activation also leads to antioxidant effects and does lead to a decrease of reactive oxygen species, which mitigates the production of senescent microglial cells. And P53 is a tumor suppressor for cancer, but it also contributes a lot to apoptosis within neurons. Apoptosis is essentially cell death, so we actually have to deactivate this within the cell so that this does not occur as much. And basically, its activation also causes amyloid beta aggregation into oligomers and fibrils, and this is really just shapes that kind of form the plaque as they stick together. So its prevention would prevent these sort of shapes from taking place. So that was kind of my overview of this pathway.